Hello students, welcome to my channel IPE Botany Hub. Let us do IPE preparation in second year botany with the most expected long answer questions from Respiration in Plants chapter. Question number 1. Give an account of glycolysis. Where does it occur? What are the end products? Trace the fate of these products in both aerobic and anaerobic respiration. Or explain the steps of EMP pathway in detail. Division of glucose into two pyruvic acid molecules is called glycolysis. It occurs in the cytosol of the cells. The various steps of this pathway worked out by German scientists Emden, Meyerhoff and Paranas, hence it is also called as EMP pathway. This is glycolytic pathway. Go through this carefully. Let us see the reactions of glycolysis. There are 10 steps in glycolysis. First step, phosphorylation. In this, glucose consumes ATP to form glucose 6-phosphate. This reaction is catalyzed by the enzyme hexokinase. Second step, isomerization. Glucose 6-phosphate isomerized to fructose 6-phosphate. This reaction is catalyzed by the enzyme phosphohexose isomerase. Third step, phosphorylation. Fructose 6-phosphate consumes ATP. It forms fructose 1,6-bisphosphate. This reaction is catalyzed by the enzyme phosphofructokinase. Fourth step, cleavage. Fructose 1,6-bisphosphate is cleaving into G3P and DHAP. G3P stands for glyceraldehyde 3-phosphate. DHAP stands for dihydroxyacetone phosphate. Both are trioses and isomers. This reaction is catalyzed by the enzyme aldolase. Fifth step, isomerization. G3P and DHAP are isomers. Among these two, G3P directly participates in the subsequent reactions. Hence, DHAP isomerized to G3P and two G3P molecules participate in further steps. This reaction is catalyzed by the enzyme triose phosphate isomerase. Sixth step, oxidation. G3P oxidizing to 1,3-bis-PGA by releasing hydrogens. Such hydrogens are accepted by NAD+, then it is oxidizing to NADH plus H+. Here, inorganic phosphate donated by H2PO4 from the cytosol. This reaction is catalyzed by the enzyme G3P dehydrogenase. Seventh step, dephosphorylation. 1,3-bis-PGA is releasing inorganic phosphate. That inorganic phosphate accepted by ADP. Then it forms ATP. Then 3-phosphoglyceric acid is forming as the product. This reaction catalyzed by the enzyme phosphoglycerokinase. In this reaction, substrate 1,3-bis-PGA is donating inorganic phosphate to ADP to synthesize ADP. Hence, it is called substrate level phosphorylation. Eighth step, isomerization or intramolecular shift. In this, shifting of phosphate from the third carbon of PGA to second carbon of PGA. This reaction is catalyzed by the enzyme phosphoglyceromutase. Ninth step, dehydration. Removal of water from 2-PGA to form phosphoenol pyruvic acid. This reaction catalyzed by the enzyme enolase. 10th step, dephosphorylation. Phosphoenol pyruvic acid is releasing inorganic phosphate. Then it forms pyruvic acid. Inorganic phosphate accepted by ADP to form ATP. This reaction is catalyzed by the enzyme pyruvic kinase. In this reaction, substrate PEP is donating inorganic phosphate to ADP to synthesize ATP. Hence, This is called substrate level phosphorylation. The end products of glycolysis. 2 pyruvic acid molecules are formed. 4 ATP molecules are formed, but 2 are utilized in first step and third step. One in first step and other one in third step. 2 NADH plus H plus molecules. Fate of pyruvic acid. The fate of pyruvic acid depends upon the availability of oxygen. In the presence of oxygen, it is completely oxidized to CO2 and H2O. In the absence of oxygen, it is converted into either ethyl alcohol and CO2 or lactic acid by a process called fermentation.